Okay, folks, if you don't mind, I think we should get going. Okay, so just before I get going, I had a couple of administrative announcements to make prior to the actual lecture material. So we're into schizophrenia again. So we started off that module. I'm not going to finish schizophrenia today. One of the things that I wanted to go back and review, because you'll need it if you're doing it as part of your presentations, is to talk to you a little bit about GWAS studies and to make sure that we're all on the same page. Some of you are in genetics and some of you are in neuroscience. And I wanted to make sure that those of you who are in neuroscience remember or recall everything that you learned about genetics, especially associated with the GWAS study that we're going to do today. And I'll come back and finish schizophrenia. The good news or bad news first? Bad news first. Okay, the bad news is the group average on the test that you wrote together as a group last Thursday, the average was 96%. Yeah, it's bad. I was expecting 100. I really was, because I was hoping that collaboratively, the more brains that you have, the better off that you would do. But I will be posting the marks, unfortunately, a little bit late. Had a lot of administrative things that I needed to take care of prior to that. There are three of you who wrote the exam at a later time. Your marks are also not up right now. They'll be going up by I believe sometime tomorrow afternoon, if I can remind Gianna in the office to do so. But you guys did a really great job, and I'm hoping that you'll continue to do a good job throughout the rest of the course. So that brings me to, at the end of today's lecture, I'm going to talk to you a little bit about your next assignment that you'll be doing, a written assignment, and then signing up for presentations on Thursday. And I'll remind you again on Thursday about this. So schizophrenia is one of those conditions, and I actually read a report as part of my administrative responsibilities here at U of T that students here at U of T will undergo 1 in 3 will undergo some sort of mental health condition, either while they're studying here, during the time that they study here, or shortly after they've left. Some of that includes things like schizophrenia. We talked about the incidence of schizophrenia being 1 in 100. That is not uncommon. That means that all of us in this room, if you think about the size of the class being roughly 60 students, that all of us will at least encounter someone that has schizophrenia sometime in your lives. We do not, even though we've been looking for this for over 25 years, we do not have a genetic smoking gun. We know that it's genetic, though. We know that if you have someone in your family that has schizophrenia or some other mental health disorder, that it is inherited. It's a heritable condition. And so we know that there's a genetic component involved. We know the genomic sequence of human DNA, and for the past 25 years, the best brilliant minds have been looking for this, but no one's really been able to find it. So I'm going to take you through this one paper, which involves a GWAS study, and I'll be reviewing a little bit of what GWAS represents. I'll give you a very brief review of schizophrenia, and then we'll take a look at maybe some of the reasons why these genetic approaches have really not been so successful. Okay, some of it is really our fault as neurobiologists, because there aren't very many neurobiologists who also study genetics. Sometimes we get this wrong idea, or we get this wrong impression. Everyone used to think it has to involve things related to neurons or BDNF, or it has to involve things related to neurotransmitter systems. But in fact, every study that 15, 20 years has actually implicated the immune system. And people used to think of that as a very bizarre, almost artifactual type of result. But I'm going to share with you that, in fact, it probably isn't. And this study here that I'm going to be taking you through actually implicates the immune system and the C4 alleles as part of the major histocompatibility complex, as part of the reason why schizophrenia is likely a neurodevelopmental disorder that manifests itself in the teenage years. And I'll bring all of that up for you again on Thursday. So I'm going to go back and tell you a little bit about what we think we know about schizophrenia as we go along. So one of our approaches in genetics over the last 30 or so years is to look through the genomic sequence to see what kind of variations exist in the genetic sequence. Now that we have fully sequenced everything since about the year 2000, as more and more individuals, and we're not talking about one or two individuals, but we're talking large numbers of individuals are now being sequenced. And in the United States, they have a 1 million human genome sequence project going on. And in Canada, we have several hundred individuals undergoing individual sequencing. We know a lot more about the genetic sequence than we did before. We know that there is a lot of variation within our genetic sequences, but not all of those variations are things that are necessarily bad for us. We know, for example, that common variants can go anywhere between 1 to 5% of all of the variations that we see in different alleles. Now this becomes important. If you have all of this existing variation already, and we know that we have so many different nucleotides to look at, and if they're all varying, how do we know which ones are associated with different types of disorders? So how do we know that one variant is actually associated with something like schizophrenia, for example? And it actually, in one way, even though in terms of technique, we knew a lot about how to sequence, it isn't until big data, big information, and our statistical power became more prevalent, and our way of looking at different DNA variations statistically became more powerful, that we really didn't understand what we were looking at. And this is where these genome-wide association studies come into play. And in some literature, as you're doing your background preparation for your presentations, in some literature it's also known as a whole genome association or a WGA. And the NIH definition that most people use is that a genome-wide association study, or a GWAS, is a study that is used to examine these different genetic variations across the entire human genome, and then to see if there's anything with those genetic variations that we can assign to different types of observable phenotypic traits. Now that's sort of the goal. But in reality, when we start doing these types of GWAS studies, it hasn't been that successful, to be quite honest. There are a number of GWAS studies that look at Parkinson's, schizophrenia. If you look at something really subtle, like reading disorders, like how do you account for whether or not someone learns to read early, can't read at all, etc. And those are really, really complex types of traits. We haven't been that successful. Technically, we've gotten better and better at sequencing the DNA, but understanding what's in the DNA has actually been more challenging than we originally thought back in the 2000s. So when we start doing a GWAS, oftentimes we look at these single nucleotide polymorphisms that I left you with before you wrote the midterm. And these SNPs, these genetic markers that vary between different individuals, are one way to look at some of those variations. So that 1 to 5% variability that we talked about. The beauty of GWAS now in the 21st century is that you don't have to look at these individually. If we're looking at 2 million differences in my genome versus 2 million in your genome, it would take a long time to scan each of those differences individually. And what GWAS does is it takes all of those different variants across a large number of individuals, and it allows us to take a look at these SNPs all at once. And that's really the major goal, is to see, are any of these SNPs that we see in my DNA or in your DNA, so somewhere between 1 and 2 million differences, are any of these SNPs or other variants, and these are harder. So if we have an insertion or a deletion, it's actually harder to pick up an insertion or a deletion than a single nucleotide polymorphism. Are any of these associated with a trait? Now I want you to be very careful with this. And this is something that I'd like you to make sure that you are aware of, even if you don't remember a lot about GWAS at the end of this. The one thing I want you to take away, and hopefully at the end of the paper that I'll present to you, is that when we talk about an association, it does not mean that whatever SNP that we find actually causes that particular disorder. Sometimes it does, but not 100%. Okay, so it's not always 100%. That is the SNP that causes Parkinson's, or that is the SNP that causes schizophrenia. So I'm going to talk to you a little bit about this. So these variations Often we isolate them. We see them running in individuals with schizophrenia at a higher statistical proportion than we do in the normal population. But that SNP doesn't necessarily mean that that is the genetic cause for what is associated with schizophrenia or Parkinson's, etc. So keep that in mind, that association doesn't necessarily mean causation. So oftentimes we start off with this null hypothesis that whatever SNP marker or whatever genetic marker that we're looking at is going to be found in equal proportions. If I have a group of individuals that have a disease or a disorder, and I have a normal population, that in theory, the null hypothesis is that that variation is going to be found equally between both of them. And whether or not that SNP is not found equally, we accept or reject that null hypothesis. And we make this basic assumption that markers are found more frequently in people that have a specific trait that we're looking for. And that's really the reason why we're doing these genome-wide association studies to begin with. So there are lots of advantages. So in the old days, so for example, I can tell you when I was working over at the CRND and I was doing basic research related to motor neuron diseases, and the lab down the hall, Dr. St. George Hislop, his lab was interested in the genetic causes that underlie Alzheimer's disease, Parkinson's, as well as ALS. And one of the things that they were doing was the old-fashioned way of doing things, where they were doing linkage analysis. And what they were trying to do was to find individuals that were affected with ALS. They would bring those individuals in, they would extract some of their blood, they would take their white blood cells, and at the family level, they tried to see if they could develop a pedigree. So they created a family tree, and they wanted to know, are there any candidate genes? They were looking for genes that were associated with things like ALS. It works great if you are looking for something that has a very easy to spot type of phenotype. So ALS is very, very characteristic. The features are very characteristic. So these types of linkage analyses were what we used to use a lot of in the 1990s and into the early 2000s before these genome-wide associations came into play. So this was great in the sense that when we started to derive a hypothesis, if, for example, we thought that superoxide dismutase, the manganese form of superoxide dismutase, was involved in things like ALS, it was easy to test. So in an unaffected individual, do we see mutations in superoxide dismutase? No. Do we see superoxide dismutase being mutated in an individual with ALS? Yes. Then it became really easy to look for a hypothesis. But we had to have a gene. We needed a candidate gene. And we know that there are lots and lots of genes. Which brings me to another question, I guess. How many genes do we actually have? 20,000? 25,000? Everyone agree that it's somewhere between 20 and 25,000. How do you know that's true? Because you read about it, you heard it in Bio 230 or something like that? Yes? But the reality is we actually don't know. And the reality is that the genes that you think about most often, we really only know and have the function for about 5,000 to about 8,000 different genes. The other ones are ones that we infer to be real genes. We don't know 100%. We think they are. We think we know what they do. But in fact, functionally, we don't know all 20,000. Functionally, we don't know all 25,000 of these different genes. So keep all of that in mind as we go along. So the GWAS study, or the GWAS, is sort of a population study. We don't do an individual or family by family per se. What we try to do is we take a look at entire populations. We look at thousands or 10,000 or tens of thousands of individuals at a time. We don't look for one gene. We look across the entire genome. We sequence the entire genome, or we genotype everything in that genome. And in theory, at least in theory, this means that complex traits can be examined, right? So complex traits meaning something very, very subtle. Things that may not be readily apparent. You might be sitting beside someone who has a learning disability or a reading disability, and you'd never know. How would you know unless you are actually testing them for reading skills? It's unlikely because all of you are here in a post-secondary school system, but those are examples of complex traits. Autism, for example, is another complex trait. There's a whole sequela of different types of phenotypes that have to be examined, and these complex traits are really good in GWAS studies, in theory, as long as you have a diagnosis that you can give. Here, it's really good because, in theory, we don't have a gene. We don't know from the beginning that gene X is involved in creating autism. What we try to do is look at individuals with autism, individuals that don't have autism, look to see what differences there are, and try to find a gene based on that. So we have no hypothesis as to a gene being involved, and this is the beauty of a GWAS study. So beyond just finding specific disorders, and in this paper in particular, this paper actually hits all 4 of these different marks. The one that I'm going to be presenting to you that came out exactly a month ago. So yes, this paper that I'm going to show you definitely tries to make an association between the C4 allele and variations in the C4 allele with schizophrenia. But beyond that, because schizophrenia has a number of different complexities, it's not just about, for example, hearing voices. It could be an individual might have issues with concentration, they might have issues with attention, whatever sort of variations that we have, this paper tries to address that. In addition, one of the things that comes out at the very beginning when I take you through this paper is they actually start off by saying, you know what, people have looked at schizophrenia before. They've looked at genes before with schizophrenia. None of that really made any sense. So we're going to try to make some sense out of this. And what they're looking at is actually a gene-gene interaction. So that if you have a change, or if you have an allele that is slightly different in the schizophrenic population, the reason why it's so difficult to actually detect that that is the gene that's involved is because it might actually work downstream on other types of genes. So there are combinations of effects that come into play, and this paper also addresses this. And then there are these gene variants, which I'm going to show you as well. So this C4 allele, you're going to see eventually that it has 4 different possible combinations. And depending on whether or not you had the C4A combination and the long combination of this, or the short, will really determine whether or not you have an increased risk of developing schizophrenia through this GWAS study. So these GWAS studies are actually really, really powerful. It's not just about finding a disorder. And again, I'll use an example from one of my colleagues, Dr. Barr, and she is actually looking at learning disabilities and reading disabilities, and what she's finding is there is no smoking gun in terms of a gene that's associated with a reading disability. It's a little bit more subtle than that. It may be a place in your genome that affects an enhancer, or it might affect something else that hits downstream gene expression. So I'm going to take you through, in case some of you are presenting any of these GWAS studies, some of the different elements in a GWAS. So keep in mind all of these different things when I take you through the paper after the break. So one of the important things is we need population level. You know, you might think that having 100 people in your study is a lot, but it's not. It's not enough statistical power to be able to get beyond population stratification. It's not enough to be able to give you significance. So we need a large number of people that have schizophrenia, for example, and we also need a good control group. And I'll come back to that a little bit later on. What you're going to see is that most studies that you look at often focus on participants who have a genetic bias toward having this disorder, or a genetic basis for this disorder. So we talked about, for example, in schizophrenia, some people develop it really early, some people develop it a little bit later. Or for example, if we're looking for Parkinson's disease and we're looking for a genetic basis, individuals that have Parkinson's at an earlier age are more likely to have a genetic basis than someone who gets Parkinson's when they're 80 years old, when their substantia nigra is already in a state of decline. So we have to make sure that we get a good working group or experimental group, and we also have to make sure that our control group is also equally well sort of balanced. Of course, the next step is to isolate DNA as well as to do the genotyping. We're really good at that at this stage. Yes, did you have a question? It's possible, 100% possible. So this is one of the reasons why you have to actually make sure that you're accounting for things in your control group. Okay, so
to make sure that they're not likely to have that disorder or disease later in life as well.
yes, that's correct. Yep, 100%. So there are different ways to handle this. So you can do what's known as a cohort study, which I'll show you, or a case control study. And some of those have these controls built in to how we do our sample selection. So great points. So right now we're really good at isolating DNA as well as genotyping. And I'll get through some of the quality controls that you should be looking at in all your papers. When you're critiquing a genomics paper, there are some things that you should be looking for and critiquing in these genomic papers, just like you would in any neuroscience paper. There are things that you should be looking for in a genetics paper. So there has to be some quality control parameters that are outlined for you, and we'll cover that a little bit later on. There has to be a statistical significance at a population level, and we'll get into statistical significance and Bonferroni corrections as we get into the lecture a little bit later on. So is that SNP that you're finding, are they actually associated with that phenotype or not when we're looking at tens of thousands of individuals? And we need to be able to have this statistical significance. The most important thing is just because we have an association means absolutely nothing. That SNP may have absolutely no functional consequence that we're aware of right now. We have to first replicate the experiment. So that hasn't been done in this paper. And then we actually have to take it one step further. We have to perform functional experiments, which this paper does do. So if you think you have isolated a SNP, a change that is important in the association with this disease that also causes that disease, then you have to do these functional verification experiments, which this group does do. And they do it very, very elegantly. It's one of the best examples in neurogenomics that I've seen in the last little while, where they've gone and done some elegant functional studies. Any questions on that before I go on? I'm hoping most of that's a review from your HMB360 lectures. Yes, no? Kind of? Good. Okay. So as Kate was asking, what sorts of things can we do? So one of the ways that we can do this, and this is the most common way that you'll read about when you're reading through GWAS papers, is to do a case control GWAS study. So you might get a group of individuals, you might go through their clinical records, and you might separate them into two different groups. You might have a case group, which has the disease phenotype that you're interested in. So we often do this here in Toronto. There are a number of groups that are doing these case control GWAS studies on autism, for example. And then you'll have a group of control unaffected individuals as your control, hence the name case control. Once we've separated these two groups out, we will then isolate the DNA, do the genotyping, and then see if there are any differences that we can look at in terms of SNPs, indels, or any other differences in the genomic material. The other way that we can do this is a cohort design type of study. So here we're looking at all sorts of different individuals at the same time. What we try to do is get people to enroll in a study, get them to all come in or all donate samples, and some of those individuals will have the disease phenotype and some will not have the disease phenotype. We think they might have it. We're not 100% sure. They come in, they give us their samples, and then we start looking at their genotype. We isolate their DNA, and then we can put them into two groups after the fact. Okay, we don't know ahead of time if they had the disease or not, but after we've isolated the DNA based on different types of SNPs that we can isolate, we might be able to break them up into disease groups versus the non-disease groups as sort of a cohort design. And oftentimes what happens is you start off with the case control and then you work backwards into sort of a cohort design study. So you think individuals might have autism, you think you've identified a particular SNP that's associated with it, you separate them out into different groups, and then you do the comparison. This particular paper that I'm going to be showing you does all of these things, plus this trio design. And you guys remember trios from HMB360, right? Trio, pretty pretty straightforward. You have the proband, the individual who is affected, the proband plus the parents, right? The parental units. And so this is a trio, and we can do the genotyping of all three of those individuals. Again, because it's a genetic study, you're going to be inheriting genetic material from your mom and dad. It makes sense to do these different trio type studies to see if there is any kind of allele being transmitted to the offspring that leads to them being affected. And again, if there's no association as our null hypothesis, it's fairly easy to determine if there is or isn't any association with the phenotype as well as the genotype, making the basic assumption that the transmission frequency is just statistically 50-50. You get one from your mom or one from your dad, and that's the reason why it should be around 50. If it's not at 50%, then we can reject the null hypothesis. So there are lots of assumptions that are made, and there's some strengths and advantages to all of these different types of things. So why would you want to do these? In fact, in an ideal world, you'd want to do all three in one study if you have the resources to be able to do this. Have any of you actually read the paper that I'm trying to present today? No? Raised eyebrows, people looking away. That's okay. This study actually does all three, and the reason why they can do all three is because they actually are spread out everywhere. They're part of a consortium. If you see the actual list of contributors, it's several hundred people that are involved in this publication. It's not just the ones that are on the front that I'm going to show you. Actually, as part of the supplemental materials, there's actually a whole page and a half of other contributors that are involved in this type of study, because they do case control, then they run it as a cohort, then they do trio studies to identify the C4 allele. And these case controls make the assumption that all of the participants are drawn from the same population. We know a little bit about that. So if you're here at U of T, the population is pretty stable. You know, everyone has they're overachievers. You know more or less that everyone's smart and you're fairly young, et cetera, et cetera. So these case control studies, we know a lot about about the population, and we think that it's the same population that we're drawing from. And we think that these case participants that are taking part represent the entire spectrum of the disease as well. And we are collecting the same genetic data and epidemiological data from the cases as well as controls. Now, the cohort studies, again, one of the advantages here is that they're a little bit more representative of the population, because we're not sorting them out to begin with. We're trying to get them all into our study, and then we're trying to see where they have to go. And then we are looking at whether or not they have the particular trait that we're looking for. The trio study, as I mentioned, is a pure genetic study. There are reasons to do this, and I'll point those out as we go along in the rest of the lecture. So some other advantages, the case control can be done in a fairly short amount of time. If you want to just see, and you have these samples sitting in your freezer, and you know where all these samples are coming from, you can do a case control study relatively quickly. And again, the cohort studies, you have to look for how these cases actually develop in your cohort. So it may take a little bit of time. It takes time for the observation to develop. The person or persons may take time to exhibit the phenotypes associated with things like autism, etc. And again, we have some other advantages in doing things like trio studies. We can go back and check for things like Mendelian inheritance. It's logistically simpler. You guys can think of reasons why, right? Why is it easier to do a trio study? If you have the proband or the affected individual coming in anyways, oftentimes it's easy to bring their parents in along with them. So logistically, you don't have to actually do a lot of recruiting. You don't necessarily have to phenotype the parents either. All you want is their genetic material. You don't really care what their phenotypes look like. And most of this is a review, I'm assuming, right? Yes, no? Some of you are looking at me like I have like four eyes, which I do because I'm wearing glasses. But is any of this completely new or completely foreign to you? It's okay to say yes. Kind of yes? Okay. I would ask you to maybe go through some of the advantages that are conferred by these different types of studies. Hopefully by the end of the study that I'm going to present to you, because it actually goes through all of these different scenarios. You'll have a better understanding of how these things actually work. I'm not going to ask you on your final exam to come up with a list of these different types of things, because that's a little boring, to be quite honest. I have better things planned for you for your final exam. But these are things that as you're reading through your paper, if you're doing a GWAS paper, you should be looking for these different elements in your GWAS study. So have any of you done genotyping? I know some of you have. Genotyping? Large-scale genotyping? How do you do it? PCR? Sure. PCR what? Okay, so you might have DNA. Where do you get that DNA from? So you don't pull it out of your wallet and say, here's my sample. Hopefully not. I guess in some ways you might be able to do that. That'd be kind of gross though. Any ideas? How do you get a sample of DNA? Really quickly without you being an expert in anything. Yes, at the back. Yep. How do you do? What sample do you use to do your DNA prep? But you don't carry cell culture, right?
No, I don't.
How about from a human sample? What sample would you be willing to give up of yourself?
Like swab your cheek or something?
Could swab your cheek. Is that a good way to do it? Swabbing your cheek?
Sure.
Sure. Okay. Confident answer. I like that. Anything else that you are aware of? Yes. So blood, sure, you could definitely do that. Yes. Collect saliva. Which way do you think is most commonly used now? So you have hair samples, which in my case, not such a good idea. You have blood samples, which require technical expertise, so you don't want anyone just drawing your blood. And then you have saliva. What do you think? Saliva? There's one other reason why you do saliva samples. Can you think of a reason why? Yes, Eddie. Yeah, you might have bacteria there. It's okay. It's easy to remove bacterial DNA. Any other reasons that you can think of? How much saliva? Like, do you walk in and you kind of spit and then you walk out? How much saliva do you actually have to give up? Not a lot. Okay. How much is not a lot? Do you want me to fill that bucket for you? You want me to fill that? How much is not a lot? Sorry? 4 swabs. Okay, so one, we don't really do cheek swabs as often. So in forensics we do, but not we, but in forensics they do. Generally speaking, in research, we don't use cheek swabs. A lot of that is dependent on what you've had for lunch. So if you've had a chicken sandwich for lunch and you do a cheek swab, you might end up having like chicken DNA samples. So we don't do cheek swabs as often. Saliva, after you've done a mouth rinse wash and then you collect saliva, is a really clean prep. How much saliva do you have to collect? Yes. You get a little tube. So it's about 5 mils of saliva. Why do you need to collect 5 mils of saliva for your genotyping? To get enough DNA? Yeah, absolutely. That's definitely one reason. Another reason? So a lot of this is actually being proactive, thinking about the future. So one day, in theory, and one of the reasons why now in research, you are actively giving saliva, you're kind of spitting into something consciously and providing about 5 mils, is because there you are providing the sample. If you think about it, in theory, if you have someone who's unconscious or sleeping on your couch, you went over and you yanked their hair or you did a little cheek swab, you could get their DNA sample without their explicit permission. And nowadays, we're thinking about those types of bioethical considerations now, because we know that at some point your DNA is going to be very private, or should be very private. So that's going sort of a little bit beyond what I need you to know. In terms of isolating genomic DNA, we get these samples, including cells that are found in your cheek, etc., in your mouth. Some of that will have DNA. We spin the saliva sample down, we collect the cells, we fractionate the cells, we break them open, we collect the DNA, we fractionate the DNA or fragment the DNA. We don't keep it intact, we fragment it into pieces. As your colleague mentioned, we will then go in and label the DNA after we do PCR amplification, and we can also put in fluorescent tags as we are PCR amplifying the different size fragments as we do this labeling in step 2. And then we have these really high density chips. So many of you took a third-year lab course, right? And did you guys do microarray analysis? Okay, how dense were your chips? How many spots were on your chip? 8,000? Okay, not bad. So here we're talking tens of thousands to 500,000, right? And it's a very dense chip. And we have on these little spotted chips, we have these different SNPs that have been identified. And every time a researcher comes out and publishes that there is a new SNP that we've identified, and we identify these sort of SNPs as RS numbers. Every new RS number that we assign can be put on this chip. It's synthesized and put on this chip. And so what we will do is take all of the samples, whether it's a cohort study, a case control study, etc. We will fragment the DNA, we will label the DNA with the fluorescent tag, and then we'll allow it to hybridize on different portions of this chip. And if it fluoresces, then that means that that region contains the SNP of interest that we have hybridization to over here on the right. And this level of fluorescence allows us to determine whether or not all of our SNPs that we think are in our sample are going to be properly genotyped. And in theory, if you have done it the right way and your labeling reactions have worked well, about 80 to 90% of all the SNPs in your DNA sample should actually be successfully genotyped within each sample. So there are positive controls that are included in here. There are negative controls that are included in this matrix up at the top, and it allows you to determine your genotype and to give you an idea of the overall efficacy of this labeling reaction. So another important thing that when you're reading through these papers, and I also want you to be aware of this just for genetics, is something known as a call rate. I just told you that reliably, you should be able to get 80 to 90%. In research terms, though, 80 to 90%, it sounds really good on paper, but it's a lot like getting an 89%, right? Nobody wants a 3.7. Everybody wants, sorry, I guess it's 84. Nobody wants an 84. You all want sort of the 4.0. And it's not quite good enough. We want a call rate, and a SNP call rate, where we know that we have functionally identified 95% of the SNPs that have been identified. We need a quality control of a SNP call rate of about 95% or higher. In theory, it's possible to get 100%. I've never seen a paper with a call rate on SNPs at 100%. Usually it's around 95% is considered like a really good genotyping, high quality research publication. And those are things that you look for. And if some SNPs are not at this call rate, and I'll show you what this looks like a little bit later on, those actually should be removed from your analysis. Most research papers, they're above this 95% threshold. Okay, so again, just sort of for completion, if you are taking a look, because we're looking at different alleles and we're looking at SNPs, for example, we might have this particular change on this allele of a T in this position versus a G in this position. If we find that the frequency of this variation is 72% of the time we see it in this form versus seeing it in this G form at the bottom at 28%, we call this the major frequency versus the minor allele frequency associated with that change. Usually it kind of runs in that 1% to about 50%. I'm going to show you why this is important when we get into the C4 alleles in a moment. And again, there are some other quality control indicators. And again, just to make this really clear, we don't know everything about the genome yet. There are some really rare alleles that we can't even detect these things. And I'll show you what that looks like when we get to the actual data in that paper. Just keep in mind that there are some that actually fall below a certain threshold. We can't even report them, but we can identify that they exist. So it's important to know how frequent is this change that you're seeing in the genome. Is that okay for everyone? I'll give you a more solid example of this when we get into the C4 allele in the paper. Keep in mind that the overall changes, the way that we see these SNPs can also be reported as a percent. Okay, so we see this particular SNP at the top 72% of the time versus down here at the bottom 28% of the time. Okay. So again, if it's possible, sometimes you're doing de novo research, you're looking for something that no one's ever described before, but for the most part, wherever it's possible, a known variant or a known SNP should also be reported along with your particular SNP that you're investigating. And this paper shows this very, very clearly, and I'll give you an example of that in that particular paper. And this is usually based on previous publications. So this paper definitely shows a previously reported SNP, and it also shows their new sort of identified variant in their particular paper. Any questions about this before I go on? You guys like statistics just as much as you like genetics, right? If you had a choice between genetics and statistics, it's really hard. Genetics, statistics, statistical genetics. Not really sure. Anyways, a couple of things to keep in mind. One of the things that I was talking to some of your colleagues about a little bit earlier, we have this sort of inherent bias in some of our populations. So for example, if we were only looking at a certain population, like the population in Europe, could we broadly apply this? Is this a SNP that's common across everyone, or is it just found in the European population? Or if we were taking a look at genetics, for example, you would think, and this would be a bias sample. Another way to look at population stratification is bias samples. If I were to ask students that were in life sciences, do you know what genetics is? I would guess that 100% of students would say in life sciences that they know what genetics is. But if I asked, is that actually a good sampling of the entire U of T population? What about all those students that are in humanities or in music, etc.? And so I've stratified the population. I've taken a bias sample from a stratified group. You are the stratified group, the life science group. And so my statistics actually have to account for whether or not I am biasing my sample or not. And one of the things that we need to consider is that when we look for tests of significance, and if you're looking for a really, really good review article, most of the data that I'm showing you is actually from Pearson and Manololio back in 2008. This actually tells you whether or not you're biasing your sample, and if you're doing a statistical correction, if you should be seeing that SNP associated with the population or not. And again, we won't worry about all of the details. It's just to give you an idea that we have to account for this population stratification. If we are biasing our sample, if you're only using samples from European ancestry and then you're correcting for it, it's a statistical sort of bag of tricks that you're using, and you have to account for that when you're doing your reporting. So this gets into this idea of linkage disequilibrium. So ideally, if things are being sorted out randomly, then things should just appear like 50% of the time. It shouldn't have a bias associated with it. However, we are sort of looking at this non-random association with alleles. If we find SNPs that are associated with the disease population, if we were to go back and look near that SNP, we might see that there are other things in that disease population that is associated with their disease condition. And the alleles of those SNPs that are in high linkage disequilibrium are usually get inherited together. This gets into this idea of haplotypes, groups of SNPs that are passed along together, and we want to know, are these groups of SNPs that we're identifying in this disease population that's being passed over to cause this disease, are they actually linked or are they not linked? Are they in linkage disequilibrium or not? And so we want to know, the higher the D value is, this linkage disequilibrium, the higher the D value is, the more likely it is that the SNP that we found is associated with the disease or disorder, and it's likely being passed along with a particular gene. Okay, so keep all of that in mind as I go through the next few slides. This idea of a linkage disequilibrium where these SNPs are being passed along together in terms of genetics is really important. And this high D value is an important one as well. This R squared statistic is actually really important, this correlation coefficient. And I'll show you an example of this in the next slide graphically. So the R squared value, it's really easy. The linkage disequilibrium, I think that this SNP I've identified is linked to some gene that causes schizophrenia. If they're linked together, should my R value be high or low? Should be high. It should be almost 100%. Right? So if they're linked together, it should be linked together really tightly, a tight correlation. It should be over 50%, random chance. It should be closer to 100. A perfect association, for example, where that SNP that you've identified in your screen is associated with a gene nearby that is also being passed along is going to cause schizophrenia, that would be like 100% or an R squared value or correlation coefficient of 1. So these are haplotype maps that you see here, or also known as LD plots, linkage disequilibrium plots. And what these are showing, the darker the red color is that for a particular haplotype block, so this is one block up here, this is another block up here, these are different SNPs that are on this particular stretch of DNA that they've been genotyping. So they genotyped this entire length of a gene or DNA, and they've identified all of these different SNPs that you see outlined here at the top. So this is one SNP, this is another SNP, all the way through. Some of those SNPs are inherited together or passed along together, and we call these haplotype blocks. And what we see is that there are some of these different SNPs that are passed along with a higher degree of affinity for each other or tighter correlation than others. And I'll just jump all the way over here. So this one over here, if we were to take a look, if we look at block number 4 and we look at the 12th SNP that was identified on this particular stretch of DNA, and this is the 12th SNP that's been identified, and we're also looking at the 16th SNP. When we take a look at how they line up on a linkage disequilibrium map, we see that together, when we look at SNP 16 and we look at SNP 12 and SNP 16, that we see that they're almost passed along together as a block, together in terms of one SNP and the other SNP at a really, really high rate, 96% or 0.96. It's almost at 100%. So if I'm passing along this SNP, number 12, then I will also be passing along almost without any hesitation, the SNP number 16. Is that okay for everyone? Yes.
Are these SNPs arranged by
On this map, yes, but in terms of numerically, no. So they're put in the order that they were identified and put into GeneBank. So some of these are not, some of these numbers up at the top here, the RS, which represents the different SNPs, are bigger numbers than the ones down here at the other end. But the way that they're arranged, yes, they would line up on a stretch of DNA. Okay. Any other questions? Yes. Sorry?
Like if you look at number 13, for
That's correct, yes. There is a threshold that you set for the different haplotype blocks. Okay, any other questions about this? Again, I'm not going to be asking you to be experts in linkage disequilibrium plots. I just wanted to give you a sense as to how different SNPs, even if they're spatially right beside each other, they don't necessarily always are always transmitted together in the same way. And so I wanted to give you an idea of how people look at these different SNPs, how we can actually determine if these are in linkage disequilibrium, which means they're being passed along together, or they're not being passed along together. Okay, so this gets into these Bonferroni corrections, and then we'll take a break. So usually in biology, we look at things being statistically significant when they are like p is less than 0.05, right? You guys have all seen papers like that. If we were doing this when we were looking at, again, let's take the lower number. So we think that in our genome, every last one of you has at least 1 million different single nucleotide polymorphisms, maybe some copy number variants, maybe some insertion deletions, whatever it is, at least 1 million SNPs. All of you have that. If I were just to compare all of you in this particular room, if I wanted to know if they were all significant, it would mean that 0.05% of this means that I would have to be examining 50,000 different SNPs, which is not a great number. It would mean that for that to be statistically significant, 50,000 different SNPs might be associated with that disease. That doesn't help me at all. I want to know if there's 1 or 2 SNPs that are associated with a particular condition. And so what we try to do is actually do what's known as a Bonferroni correction, which is taking the level of significance at a population level. If you've ever seen a Bonferroni correction, this is what a Bonferroni correction actually represents. And this alpha prime is the significance level. We can still set the significance at 0.05. We could set it at 0.01. Again, more stringent if we wanted to. You have to be told what your Bonferroni correction thresholds are. But let's say we're using 0.05 in our Bonferroni correction. If we're looking at the significance level at 0.05, and we've examined 1 million different SNPs, that would mean for one SNP to actually be associated with the disease among all of these different SNPs that are being identified, it would have to pass a threshold value of being over 5 times 10 to the 8th, 10 to the minus 8th. Okay, so this is how we get a Bonferroni correction. In order for that SNP to be significant, it has to be higher than this number here. Again, keep that in mind. Any questions about this before I go on? Why is that important? Because usually what happens is when we sequence the entire genome, chromosome 1, chromosome 2, chromosome 3, 4, all the way down to chromosome X, for example, what we do is we set this threshold on this type of plot. And for those of you who took this with me before, what is this plot known as? Genomics people, what is this plot known as? I think I heard someone say it. If you go to New York City, you look at the skyline. What skyline do you look at? I think it's time for a road trip. We're all going to Manhattan. This is sort of a Manhattan plot because it kind of looks like the skyscrapers, etc. On this Manhattan plot where we're looking at all of these different SNPs. These are all different SNPs, all lined up here. 1 million of these SNPs are all lined up here on the bottom. If we're looking to see, is there any particular SNP that is associated with rheumatoid arthritis in these 1,522 cases, and we're looking at all of these different SNPs that are being lined up, is there anything that really is above this line of statistical significance, this Bonferroni corrected statistical significance that you see here? Out of all of those millions, of all of those different SNPs that we have down here, only a few do, which is why we set that Bonferroni correction. We're not looking at 50,000 SNPs. We're only looking at 2 or 3. And this is the reason why we do these Bonferroni corrections for statistical significance. Okay? So we'll take a break now, if that's okay, and then we'll come back. And I think we're going to end a little bit early today, and I'll go through what you need to do for your upcoming assignment. a couple of other things just to mention to you. A really, really good GWAS study, if it's possible to do so, will always have a follow-up. This particular paper that I'm going to present actually does have a follow-up to it, which means that there has to be some sort of functionality associated with whatever you've identified in your GWAS screen. Just before I get there, to clear up a couple of things for you. So this plot is generated from genotyping thousands of individuals. In this case, 1,000 cases with arthritis, 1850 with controls. They're all lined up there on the bottom. All of their SNPs are lined up there on the bottom. So there are all sorts of different variations amongst their genotypes. So it's just like you, if I were to compare myself to any of you, so if I were to compare myself with Jenna, for example, just as a small example, she probably has differences because she has brown hair, I have black hair, so there might be some differences there. There might be some differences in height that are different SNPs. But let's say we're actually looking for the gene that's associated with myopia, like having nearsightedness, if there is a gene associated with that. We would have to line up all of our differences and then see if there are any that kind of pop out above this threshold, which is that Bonferroni. None of this is done manually. It's all computer generated. All of these data are computer generated. The Bonferroni correction you set. So this is a log to the -10 scale here, and you can see that this threshold is pretty standard at about 7. Log 10 to the -7, in other words, and this is actually a really high threshold to see if there's any significance. So we would do all of those things, and let's say we've identified three different SNPs that are associated with it. And let's say we think that some of those SNPs, they don't always have to be, but let's say we think some of those SNPs that we've identified or something near those SNPs are actually responsible for rheumatoid arthritis. Then we would have to go in, look for that particular gene if we have one. In this case, they're lucky, they do. If they have that gene, or if that SNP actually itself confers some sort of disease risk. There are a few examples in the literature. If you are looking at warfarin and your ability to break down warfarin, if you have one SNP versus another, you will metabolize warfarin at different levels, and it can be actually a dangerous situation. You can identify that right away. That SNP is there. That SNP confers a difference in your cytochrome P450, and there is no other candidate gene. That SNP, if you have it, you have you are either a fast or a slow metabolizer of warfarin. But you'd still want to go back and check in mice. Is this SNP that causes the single nucleotide polymorphism, is this what causes me to be more susceptible to bleeding after taking warfarin, which is an anticoagulant, or not? And we could confirm this in cell culture. We could do knockout mice. We could knock in a gene, for example. We could do CRISPR-Cas9, since that's the flavor of the year. But we would have to do some sort of functional study. So again, other SNPs or other GWAS studies where we've had some roadblocks, they go through all of this stuff. They've just gone through, they've done the genome-wide association study, they've done it on tens of thousands of individuals, and when they go back, the little SNPs that they've identified, they don't actually sit near any particular gene. There's no association. Could be in an intron region. It could be in something that no one's ever reported before. So sometimes these GWAS studies identify regions that have no known predicted genes. Well, is that an issue? Sometimes, yes, because those might actually be pseudogenes, or they could be genes that we don't think look like normal genes. Can you think of any examples that you are aware of that you've learned about related to like pseudogenes or non-canonical start sites?
That one gene from, what was it called? I don't remember.
Thanks, Jeff. That was really helpful. Anyone else? So for example, FNDC5. That actually is a gene, though.
But it has a non-canonical star sequence.
No, it does not. So I think what you're talking about is Irisen, which has a non-canonical start sequence. It doesn't have an ATG sequence. It has an ATA sequence, which is non-canonical, but it still exists in theory in our genetic sequence. So is it an issue? It's something to be aware of, that sometimes our GWAS studies report back a SNP that doesn't fall into a normal predicted gene region. And sometimes replication in a larger sample that we always want in science, sometimes replication isn't possible. You might be dealing with a really, really rare condition, and you've identified a SNP, and maybe there are only 200 people in the entire world that have it. So we'll see an example of this a little bit later on in the course. So as far as we know, in the entire world of several billion people, we know of only about 50 and probably members of their family that carry a mutation for fatal familial insomnia. They literally kill themselves by not being able to sleep as soon as they reach the age of 40. Can you do these types of large-scale GWAS studies? No, you can't. But then again, we don't need to in that particular case. Simple genetics is enough to determine, we think, what might be the issue in those individuals. So it's not always possible to do a larger sample for really, really rare diseases, things like FFI or fatal familial insomnia. So this is the paper that made all the big news. Why is it such a big news? Or why is it such big news? Actually, there are a couple of reasons that I'm going to go through. One, it's a very, very comprehensive paper. Second, it really ties in everything that we know about the phenotype associated with schizophrenia. Third, it also fits in all the genetic information that came out in 2012 from Kim McAllister's group that also was the first to identify that a major histocompatibility complex, something in your immune system, might be underlying schizophrenia. No one was able to, for the past 4 years or 3 years or so, identify what that was. Like, it doesn't make any sense. Why is the immune system and something that's associated with your major histocompatibility complex, why is that the issue in individuals with schizophrenia? It had never been really thought of. But this paper actually ties in what we know about the physiology, the pathology, the timing of events, some of the changes that we see in the brains of individuals with schizophrenia. All of it is tied together in this one paper. As I mentioned to you before, this looks like a really small list of authors. However, if you go through the schizophrenia working group in the Psychiatric Genomics Consortium, that actually is a huge number of individuals. So this is not a single site study. This is a multi-site study. You can also see that it took a long time for them to publish this work as well. It was submitted back in the 19th of May in 2015. It only got accepted in the middle of December. So they had to do a lot of follow-up studies, and I'll take you through what some of those studies are. So some of this is from what we covered at the end of last week, just as a review for you. Kim McAllister's group in 2012, 2013 actually identified a really, really strong, I mean, it was blaring. It came off the Manhattan plot like you wouldn't believe. Everyone was able to go back and verify, yes, you know, Kim McAllister, you've identified a risk SNP associated with schizophrenia, but we have no idea what this means. We don't know where it sits, or it's just there. It's sort of like a flag. You put a flag on the top of the Sid Smith building and say, somewhere in this building, you're going to find Dr. Vervaeke wandering around. Well, maybe you will, and maybe you won't. A lot of this is related to the fact that there are a large number of loci that have these different SNPs that associate with schizophrenia. Lots of them, not just this one that Kim McAllister found. These loci, we don't know a lot about them. Again, most of them are immune. We were sort of blinded by the fact that it has to be something related to the brain. It has to be BDNF. It has to be something related to a structural protein. It has to be a receptor. That's the only reason. It has to be dopamine, because dopamine is at fault in schizophrenia. And for the longest time, these functional alleles kind of blinded us to the fact that maybe the answer actually was right in front of our faces on the chromosome 6, where it was identified very strongly that in this large section of chromosome 6, which is what we normally use for our immune systems, that maybe the mechanism for schizophrenia was actually there. So again, we don't know, because this schizophrenia major histocompatibility, or MHC association, has been really, really hard for neurobiologists and even immunologists to get a really good handle on. And part of this is, if you were to take a look at the mapping, and this paper actually shows you some of it, if you look at the mapping in this 100 megabase pair region, which is a large stretch of this chromosome, chromosome number 6, there are hundreds of different genes that sit in this region. So our job when we're looking at genome-wide association studies is to try to narrow things down. We set the Bonferroni correction so that we don't look at 50,000 genes. We look at a couple of genes that are statistically significant. But in this region where we see lots and lots of variation, and we see this single nucleotide polymorphism that just jumps out at the page, it doesn't cause the disease by itself, but there has to be something in that haplotype block that's associated with schizophrenia that gets inherited. Was completely unknown for the last 3 years. Kim McAllister's study replicated, but never really validated. How do we make sense of this large region that is in obvious linkage disequilibrium? Something is being transmitted with this SNP that she identified in schizophrenia. What is it? We don't know. It's like looking for a needle in a haystack. So these authors actually decided to do a semi-logical approach. So they looked at this entire stretch. So we know a lot about this stretch, this 100 megabase stretch in chromosome 6. And they looked at the literature, and they did a thorough literature search, which is a great way to actually begin these types of GWAS studies. And in some papers that started to come out after Kim McAllister's study, it suggested that there was an association of schizophrenia to what is known as the CSMD1 gene, which is the Cub-Suchi multiple domains 1 gene, that was associated with different forms of schizophrenia. Totally different chromosome, by the way, but it was still sort of associated with schizophrenia. Now, why is that important? Because the CSMD1 is actually regulated by the C4. So even though this CSMD1, which has been implicated also as a totally different SNP, totally different mutation on a totally different chromosome, because Kim McAllister's group was saying, like, you know what, there's this big massive signal coming out of chromosome 6. Doesn't make any sense. We don't know where it is. But if we look around at other chromosomes, maybe something in chromosome 6 here affects other chromosomes that are nearby. And one of those is on the C4 gene, which is in chromosome 6. So the C4 gene regulates CSMD1, which is on a totally separate chromosome. So they went with the idea that maybe we should narrow things down. Maybe we've been looking at it the wrong way. Maybe instead of trying to brute force things, maybe we should start going backwards, seeing what other genes are regulated by what's in this stretch that we've identified in chromosome 6. One of those potential genes is the C4 gene. Now, when you look at the C4 gene, one of the things that's really interesting about this is that you can actually have multiple copies of the C4 gene. You can have anywhere from 1 copy to 4 separate copies of the C4 gene on each of the copies, one from the maternal side, one from the paternal side of chromosome 6. To make this even worse, though, is that the human C4 gene exists as 2 functionally distinct or paralogous genes or isotypes. So 2 different isoforms, if you want to think of it simply, of the C4 gene. C4a as 1 isotype of a gene, and C4b. And again, there's a lot of variation in structure as well as copy number of these genes as well. So if this is indeed the gene that they're looking for, this gene has a lot of complexity that's built into it. So let's assume for the time being that Kim McAllister was right. There's this big flag going up that somewhere around this flag, this single nucleotide polymorphism that I identified way back in 2012, when this group comes in, they want to see what's around this flag that might be related to this particular gene that regulates other known genes associated with schizophrenia. It's this C4. But once they start looking at it, they go, This is going to be more problematic than we thought. There are so many different isotypes that exist of the C4 gene. And they also target many, many different things. Now, this is what the C4 gene looks like. So here is the C4 gene sitting right here on chromosome 6. There are hundreds of genes that are around it. The gene that, the SNP that was identified by Kim McAllister that was blaringly hot was way out here, not on this particular map, but something was being transmitted along with it. That SNP by itself did not cause schizophrenia. People went back and verified it. Something in chromosome 6 has been causing schizophrenia that hasn't been identified yet. So if you take a look at this C4 gene, that actually exists as a C4A, which is this copy here, C4A, where you see these variations at the top. So C here and a G here, T here, G and C. And that's one variation, that's C4A. C4B is down here, so it's a different series of SNPs. So we have a single nucleotide polymorphism here at T. This is also a variation here at C. This is another highlighted single nucleotide polymorphism. So we have two different variations of this C4 gene. That's one element of it. The other element that makes this even more complex is that we can either have a long version of the C4A or a short version of the C4A. So we have four different types. We have long version C4A, short version C4A, long version C4B, and then short version C4B. Now, what makes this even more interesting and more complex is that sitting right here between exon 9 and exon 10 is this insertion that makes this gene longer. So this insertion is this HERV, which is actually a human endogenous retroviral vector-like sequence or virus-like sequence. Do any of you remember endogenous retroviruses? Yeah? What do you remember about endogenous retroviruses in the nervous system? So one of the things that make it really interesting is when these endogenous retroviral elements get inserted in different intronic regions, they can actually help to regulate the function of other nearby genes. So here is the C4A, or either the long form if you have the human endogenous retroviral element put into this intron. If you don't have it in the intron, this is the shorter version that gets generated. So we have four different structural variations that are possible from this C4A. Now this is probably the reason why these authors suggest why when people looked at this originally, that because of the tremendous amount of variability in the C4 gene, because we have four different gene elements right here, that there wasn't a really strong association made originally by Kim McAllister back in 2012. So they want to look at this in more detail. Is this really the reason why we get schizophrenia? So yes, there was a flag raised. This is in the region of the flag that was raised earlier. We have lots and lots of variability. We know that C4 regulates the CSMD1 gene that's been implicated in schizophrenia. There's lots and lots of reasons why it couldn't have been detected earlier because there's so much variability. Is this really the reason why this is so important? Let me again just ask you a really quick question because it was kind of an interesting exercise that I did with students who are dropping by the office today. How many of you, yes, Anne? Yeah? Yeah? It regulates.
Because on the slides it says CSMD1 includes regulator.
I'm gonna have to go back and check, Anne, but the way I remember it, and I'm not sure if that's the way I wrote it, and I'll have to go back and check, is that it regulates the function of the CSMD1. So I'll get back to you and correct that if it's wrong, okay? Which wouldn't be anything new. How many of you have done PCR before? Okay. How many of you have ever heard of droplet or digital droplet PCR? Have any of you heard of it? Have any of you done it? Have you done it, Ursuline? No? Any of you? No? Can you imagine what digital droplet or droplet digital PCR looks like? Not really, right? Can you actually envision like PCR? So one of the things that they did was, from a very, very small sample, what they wanted to do was to see if they could expand and see what the relative contributions of all of these different versions of the C4 gene looks like. So there's C4A, long, short, with the HERV in it or not. And in trio studies, so they took the parental and then they took the proband individual that was actually affected. When they did these types of studies, they used digital droplet PCR, which is a very, very sensitive way to do this. So what it does is it actually expands a PCR reaction, and it separates them out into little droplets, and you see the PCR reaction going on, or you look at it in real time, and you can actually see the different PCR products that are separated out from that one sample into different droplet samples, some being amplified more than others. And so you can actually compare in a sample, how many copies of C4A do I have? How many copies of C4B do I have? How many copies of the long version, short version in that one sample at the same time? I don't have to run 4 separate samples. That one little sample that I prepared actually allows me to examine all of them at the same time. So digital droplet PCR is actually a great way to look at copy numbers within a small sample that you start off with. You don't have to run 4 different ones anymore. In fact, you can run multiple versions of this, and it's one of those techniques that is gaining in popularity. Of the 25 or so students that dropped by the office today that I asked, about 2 of them actually mentioned that they use digital droplet PCR, which I thought was pretty remarkable that undergraduates are actually using this, because the machine itself is fairly new. It's only about 2 years on the market, and if they're using it here at U of T, that's pretty darn good. So I told you one of the things that we look for is what is the major allele frequency, what's the minor allele frequency, do we have like really, really rare versions of this? And in fact, we do. If when they do these digital droplet PCRs across all these individuals in those trio studies, mother, father, and then the children, when they take a look at the overall frequency, the vast majority of them have the C4A long version, coupled with the C4B long version, which is what you see here, about 41% of the time. 31% of the time, they have the C4A long version and the C4B short version. And so these structural isoforms were being piled up. They were getting information about what is the baseline value in the European population, which they started out with. So they were stratifying their sample. They had a very small group of individuals that they did these studies on, European ancestry population, before they expanded their study to GWAS. What is the overall frequency at which we get these different types of variants in the population? So yes, they saw all sorts of different structural variations in the C4 gene, which again, you don't have to memorize all of these numbers, nor do you have to memorize the more common one, but this is the way they started out. They wanted to know, is there variability? Yes. Which isoforms are the ones that are the most commonly found variants? It turns out to be the ones that involve the C4A, along with other variants associated with it. And overall, they found that both C4A and C4B vary in copy number and whether or not they have the long version with the HERV or the endogenous retrovirus or not. And again, just to keep in mind, these retroviral insertions have been known to enhance or modulate the function of other types of genes. It might also affect its own expression as well. And so one of the things that they wanted to know really quickly off the top, if there is a long version that has this endogenous retroviral enhancer that's associated with it, does it actually affect whether or not the C4 gene gets turned on more or less? So they also wanted to know, like, how does this affect RNA expression in different adult samples, including those that had schizophrenia versus those that didn't have schizophrenia? So what they found was the RNA expression of the C4A variant, the one that they found most frequently anyway, at the very top in that particular chart, the C4A. One of the things that they found in this particular study is that the C4A and C4B increased proportionally just as they saw, like if you have more copies of C4A or more copies of C4B, you'll get more in your serum. The expression level, though, of C4A tended to be about 2 or 3 times greater than the expression levels of C4B, which was surprising. So yes, the frequency is higher, but when they went back and looked, they were actually able to see that the RNA levels were different than they sort of expected, that the C4A levels were much higher than they were able to correct for. And one of the reasons why they think that this happens is if you have that HERV sequence that was inserted in there, it increased the likelihood that you would be increasing the levels of C4A. So this might account for the differences in the C4A, the frequency that they saw, the 41%, and the overall expression levels of C4A being higher if you have the long version that contains the endogenous retroviral sequence. So again, using that PCR, I won't go through all of the details, but they were able to show that overall, as you increase copy number, so if you have no copies, 1 copy of C4A, 2 copies of C4A, 3, 4, etc., that you increase the overall RNA expression level. Same thing for C4B. 1 copy, 2 copies, 3, the more copies that you have, the higher the RNA expression is of C4B, as on that previous slide. But if you have the endogenous retroviral sequence inserted in, that actually enhances C4A versus that of C4B. So something in that endogenous retroviral vector increases the likelihood that C4A is going to be increased. So it seems to be that C4A is turned on when we have this long version, the one that has this endogenous retrovirus. So then they started taking a look. Okay, so we have all of this anecdotal evidence. We started off with Kim McAllister saying that there is this sequence somewhere in chromosome number 6 that is related to the major histocompatibility complex. We just identified that overall that there's a lot of variation in the C4 gene, which may affect the expression of other genes or be affected by the expression of other genes. And we think that this might be related to schizophrenia. We don't know for sure. We know that there's a lot of variation. We know that whether or not you have the long version or the short version will determine the overall expression. So if you were to take a look, can we see if in schizophrenic individuals, not just in European ancestry, but across multiple centers, can we do this genome-wide association study from 22 different countries and see if there is an association built up around the C4A? So again, they're using a lot of different individuals. So they have a case control study. So they have about 30,000 schizophrenic cases and about 36,000 controls from about 22 different countries. They are looking at the association in that region, not the entire 1 million different SNPs, but just in the region of the major histocompatibility locus on chromosome 6. When they take a look at this, do they see any sorts of associations based on this? Again, just to keep things in perspective, this is that original SNP that was identified by Kim McAllister. It's the highest one on here. It has no known function, and this is that RS131940504. So they are doing exactly the control that I told you about earlier. Always include as a control something that's been published in the literature. They do. They also see it blazingly hot. But one of the things that they see is when you start looking at individuals that have an increased schizophrenia association, and they're using the same sort of scale, cutoff scale that we looked at earlier, when you start looking at this, then they see a clear association with CA4, but not with the C4B. So in schizophrenic individuals, they are seeing that the elevation of this particular isoform of C4A is actually very strongly associated versus the C4B in schizophrenia. And it's pretty conclusive evidence. So they're able to narrow down in this entire region, this one region that they are identifying here in chromosome 6. Okay, so it suggests maybe, and I'll go through this again with you on Thursday to just wrap up this paper and then show you the functional studies, which are conclusive, very, very, very conclusive evidence that this is sort of a really tightly associated SNP that seems to regulate the features associated with schizophrenia. So this is that association peak that I showed you at C4. What this point is showing is that there is a cluster of other SNPs that are around this particular originally identified SNP, this RS131. So there are some other SNPs that are there, but there is another peak of association that I showed you centered around that diamond C4, and that's that C4A. And there is a very strong correlation that they see statistically with that particular SNP. And now they have to just show, is there actually a functional consequence of having C4A expression? And this is sort of where I'm going to take you in the next hour on Thursday. So let me just answer a couple of questions about what you need to be doing in the next little while. I'm hoping that most of you are doing okay with your group writing assignment. It's about a page and a half, 2 pages-ish. You guys are doing okay? Not sure? You get started on Monday, Sunday, Saturday, sometime? Okay. So this is going to be due next week at the start of class. So you'll drop off a hard copy. You'll have all the information on it. Just a reminder that on Blackboard, they're not available right now, but they will be as of 8 p.m. on March the 3rd, which is this Thursday. I'll remind you again on Thursday's class that you will sign up, or one person from your group will sign up to present on March the 7th. So you'll put your name down if you want to present on March the 17th, on March the 24th, or on March the 31st. Just as a show of hands, how many of you are thinking of presenting on March the 17th? Okay, March the 24th and the 31st. Maybe it'll work out. Okay, it might work. Okay, not bad. So we're going to take a maximum of 6 presentations a day. We'll be separating out into 2 groups. I've asked the TAs, and I'm going to be working with them to make sure that when they're going through the rubric, that we're standardizing the rubric for everyone. We want this to be a fair and equitable experience for all of you. It's not, one of my colleagues likes to have all these sort of bells and whistles in their presentations. I could care less if you like move your hands around. Could care less. I could care less if you're a razzle-dazzle speaker. Great if you are. But it's not about how well you present at this stage. Most of you will have had some experience presenting, but it's to give you more experience presenting, and it's more about the content and how you answer questions and how you prepare your slides and how you edit your slides than the actual presentation delivery. If you can do handstands, if you can do cartwheels, if you want to bring coffee and tea to your TA, go for it. Not going to get you any extra marks. But it's really about the content and how you answer questions. I'm going to also ask you, unless you have a very, very good reason, 1% of your mark is going to be there for you showing up to the 2 other presentations. I think it's just really professional if you show up to the other presentations so that it's not an empty room on March 31st where Lauren is presenting in front of her TA only. Right? It'd be great if you are still coming and supporting her in her group on March the 31st. So you'll have like nominal grades associated with it. I am going to ask the TAs to have a little sign-in sheet that you'll sign in just to make sure. And all you have to do is show up. You don't have to answer questions. Don't have to ask. I'm hoping that you get excited by what your peers are presenting, that you will ask questions, but there's no onus on you to do that. Do you have any questions about this before we wrap things up? No? How many of you, just again out of curiosity, because I wouldn't mind sending a couple of you to do this, how many of you would actually like to see from start to finish how you do genotyping in something like autism over at Sick Kids? Like, it takes about a couple of days. Okay, so maybe you guys can email me and then we'll set you up. We'll send you over to Stephen Scherer's lab. You can go through the whole process with them from saliva samples all the way through to how they do the genome sequencing. And it'd be kind of an interesting exercise to hear what you guys thought about the whole process and what you gathered from lecture and what sort of stuck with you from actually seeing it. Okay, so we're going to end a little bit early. Please be careful on your way home. I don't know how much it's snowing outside, but I'm hoping it's not a terrible commute if you are traveling.